All right, welcome everyone. Thank you for logging into our webinar today. I hope you can hear me clearly. If not, just let me know in the chat. My name is Tamás Kis, and I'm a research fellow and physician at the Semmelweis University. And it's my honor to announce that we start a new webinar series on applied bioinformatics, sponsored by our partner, Data Science and Me, LLC. So the first presenter of our series is Teadora Tyler from the Department of Anatomy at the Semmelweis University, and she was give us a friendly introduction to the fascinating new world of single-cell sequencing. The presentation will be approximately 25 minutes long. At the end, you will have a chance to ask questions via the Zoom chat, and the presentation will be recorded and we will make it available online later this week. So, Teadora, please.
Thank you. I also want to welcome everyone. Thank you for joining us today. And I also want to thank the opportunity for having me today. And yeah, let's get straight to it. I think we can all agree on that biology is amazingly complex, and we are just beginning to understand that complexity. Whether we are talking about cell culture, an organoid, worm, or a plant, or a mice, or a human being, they will all be made up of different cells. And these different cells will contain vast amounts of information on genomic, transcriptomic, epigenetic, and proteomic levels, all to explore in order to understand their biological functioning, not just on their cellular and molecular level, but also on a network level, on the level of the organism. Today, we will be focusing on methods to explore the transcriptomic information in these cells. To do so, new generation sequencing techniques allow us to take a deep look into the RNA content of a cell. And in any case, there are different techniques, but in any case, it will involve the extraction of the RNA from the cell, the fragmentation and the reverse transcription of those RNAs molecules, and PCR amplification, and finally the sequencing of those fragments. Amplification is important because in each cell there's only a miniature amount of RNA, and so in order to be able to analyze them, we have to amplify them. Bulk techniques have been around for quite a while. I'm sure that many of you know them. And here I just want to demonstrate quickly the difference between bulk and single-cell RNA sequencing techniques using the famous smoothie analogy. Imagine that you have a fruit bowl. You can decide to put all your fruit in a mixer and you will have a really nice smoothie. However, if I go up to you and ask how many blueberries were put in that smoothie, you will have no precise way of answering that question. However, if you decide to make a fruit salad out of your fruit, then you will have an easy task. You just have to count the blueberries. And if you have some time, then you can also organize them based on some certain traits. And this is where lies the power of single-cell RNA sequencing, because while bulk technologies give you information on average from thousands or millions of cells, single-cell techniques allow to do that at the level of the single cell. And that to put that in a scientific context, just imagine that the fruit is a mouse brain, and the different types of fruit is the different types of cells making up that brain. So how does that actually work? Today, I will only be focusing on the most widely used and one of the most best documented technique from the 10x Genomics company, which is a droplet-based microfluidic technique. So after the target tissue has been appropriately dissociated, the cells or nuclei will be introduced or loaded onto a microfluidic device in which each individual cells will be joined together with distinctly barcoded beads into oil droplets. So ideally, in one oil droplet, there will be only one individual cell, and one distinctly barcoded bead. Now, these beads also contain PCR reagents. So these droplets can be viewed as individual little micro environments for individual reactions. And what are these beads? These beads are the key to achieve the single cell RNA sequencing, because each bead has distinct unique oligonucleotide sequences on their surface. And what I want to highlight is that within this sequence is the 10x barcode, which will serve as like a cell ID in every downstream step. So this will make it possible to trace the transcripts back to their original cell. So after this has been done, reverse transcription and PCR amplification will take place. And after certain delicate steps, the library constructs will be sent for sequencing. And at some point, you will get back your data and you can dive into the data analysis. If you're interested in more detail, then there are many useful videos available, one of which is referenced here. Just a few useful information. The 10x library constructs are compatible with Illumina sequencing platforms. And this is just mentioned because for me, it wasn't always clear how these constructs are made up. And what I want to highlight is that when you receive your data, each transcript will have 2 bits of information associated with them, the read 1 and read 2. And very basically what you have to keep in mind is that read 1 contains the cell ID, the 10x barcode that we just discussed, and the unique molecular identifier, which is used to identify each transcript found in each cell individually. Read 2 will contain the target transcript that you're interested in, and the sample ID, which sample was that transcript detected from. It's also good to know that choosing the right approach that you will apply for your sample also depends on the sample type. So very often you will be working with intact live cells, in which case you can proceed with regular single cell RNA sequencing protocol, but very often we work with frozen tissue. In this case, you have to be aware that the cell membrane isn't necessarily intact, and so the only thing you can surely use is the nucleus. So your protocol will have to be adjusted accordingly. And of course, many times we work with fixed tissue or paraffin-embedded sections, and 10x also offers solutions for such cases. But these spatial transcriptomics and high throughput in situ solutions are not exactly single cell RNA sequencing. However, in 2021, the single cell sequencing of fixed tissue has also been performed successfully. So I'm sure that in a few years, this will also be a benchmarked protocol. And another important information is that your target cell type's size is also important because, for example, some cells are just too big to fit into the platform of 10x. So for example, in cardiomyocytes case. So in that case, you have to choose a different platform. Luckily, there is a vast amount of information available on the 10x Genomics support page, so you can browse whatever question you have there. If we take a look at literature, we can see that almost all areas of research use single cell RNA sequencing in some context. Because the quantification of gene expression and the level of single cells is just extremely useful all the way from discovering new cell types, genes, or variants, splice variants, through translational science opportunities, testing new drug targets, all the way to clinical use, for example, with testing therapy response. And to take a look at some examples, oncology is one of the fields that uses single-cell RNA sequencing the most frequently and with most clinical relevance as well. And the good news is that oncology has a really well-curated atlas of cancer cells, which today counts more than 2 million sequenced cells. And all of these data are available for analysis for whoever is interested. The same goes for immunology. Immune cells are notoriously heterogeneous, and single-cell RNA sequencing is a perfect tool to explore that heterogeneity. The Immune Cell Atlas, Human Cell Atlas, today counts more than 1 million cells available for analysis. And of course, I have to mention neuroscience, because the brain cells are also extremely heterogeneous. And I want to highlight two projects: the Brain Initiative, which aims to integrate morphological, electrophysical, and single cell data together to provide a comprehensive database of brain cells, which will probably prove really useful for neuroscientists from all areas. And the other thing is the Allen Brain Institute, which has been working with single cell sequencing from the beginning mostly. And they have online platforms which are really useful in the everyday research life. So I also use them, and they have been proved very useful. I can only recommend them. When someone starts to plan a single cell RNA sequencing experiment, there are some parameters that they have to be aware of. And one of these are the number of cells per sample that they want to recover. This can range from a few hundred cells to 10,000 cells in the case of 10x workflow, but it's also influenced by a lot of factors, for example, money, and of course, tissue quality and the platform of sequencing. The more you want to sequence, the more it will cost, of course. The other parameter is sequencing depth, which means the number of raw sequencing reads that you will recover per cell. And this can range from the minimum of 20,000 to up to 150,000 per cell. And this also can greatly influence the cost of sequencing, because imagine if you have 6 samples and 5,000 cells per sample sequenced at the 50,000 reads per cell, that's a big number. So you have to be aware of these parameters. But how to decide between them? You really have to be clear about your research goal, what you want to answer. Will you be needing to identify rare cell types? Because in this case, you will need a lot of cells to be sequenced in order to find those, recover those rare cells. Will you be comparing groups? In which case, you will probably need a higher sample number, so your cost will be initially higher, and you will have to take that also into consideration. Or will you probably need to identify lowly expressed genes as well, in which case you will have to invest in sequencing depth to recover those genes, detect those genes. And of course, your funding will also help to decide whether you want it or not. So how much does it cost? What we can say for sure that it costs a lot more than bulk RNA sequencing. And just an example accessed at an online platform of Core Facility a few days ago. The most widely used 10x approach here costs almost $3,000 for the first sample, and almost $1,600 for each additional sample. So this can give you an idea about the magnitude of the funding needed. So once you manage to do all that and get your data, let's take a look at how data analysis takes place. The very first thing to do is the quality control of the raw sequencing reads, which at this point looks quite scary, but we don't have to be afraid because the service provider usually takes care of these steps, together with aligning to the target genome and constructing the matrix of the detected genes in the rows and the detected cells in the columns, making up a gigantic matrix filled with the gene expression values. And this matrix is usually what you start to work with. All your desired downstream analysis, which you can conduct in Python or R. And this reference here is a database of all tools, and there are a lot of tools available. So once you have your count matrix, the first step is to filter your cells based on quality metrics, because you only want to include cells that you are very sure that are good quality and you can rely on them. Once this is done, you have to make your cells comparable, because each cell can have a different sequencing depth, and so they are not directly comparable. So you have to scale and transform your gene expression values in order to analyze them together. And once you did that, you still face the problem that you have enormous data with a multitude of variables, and somehow you have to highlight the important patterns within that. And that's where dimension reduction algorithms come in. Very often you will have to use a principal component analysis, which does basically that. It brings out the strongest patterns of your data, and once you did that, you can move on for the clustering, which is probably the most fun step, because you can produce these very famous plots that you see in every second publication nowadays. These are plots that aim to visualize your data, your multi-dimensional data in a reduced dimension space. And the important thing you have to understand about them is that one point in such a plot means or refers to one cell. And cells are placed in this space based on their the similarity of their gene expression. So ideally, cells will cluster together based on their similar gene expression, and distinct different cell types or clusters will be separated. And once you have such a structure, you can, for example, project the expression of known marker genes on this structure. And for example, here I have brain cells and oligodendrocyte. An oligodendrocyte marker is depicted with the expression of an oligodendrocyte marker is depicted with red, and it really clearly helps define the cluster of oligodendrocytes. In this way, you can identify your clusters, annotate them, and go on with whatever analysis you need to answer your question. Very often, you will want to compare the gene expression of certain groups, or really do whatever you need to answer your questions. For example, you can try to put your differentially expressed genes into a more functional context to be able to understand what those changes could mean, what they could, what sort of functioning they could affect. Or another interesting thing you could do is to try to identify the transcription factors that govern the genes that were differentially expressed between your groups, which could really put another layer of information on your data, and also a great opportunity to make really fancy plots. And the last thing that I want to highlight about single-cell RNA sequencing is that it gives you an opportunity to explore cellular communication patterns, cell types, in a cell type specific way. Because once you annotated your clusters, as I showed here, you can use those clusters and analyze their known ligand and receptor expression, and predict the communication, the potential communication between each clusters, which can give great insight into a more network level understanding of how, for example, the brain works, or how whatever tissue is our target could work. And that is all I wanted to say. If you have any questions, please put it in the chat, or feel free to email me and we can try to answer your question. And once again, thank you for being here, and thank you for the opportunity to present.
Thank you, Teadora. So if you have any question, please feel free to write to the chat box of the Zoom, and I think Teadora would be really happy to answer it. If you don't have any question, or you will have question in the future, just write down Teadora's email address, and you can reach her anytime.
I also put it in the chat to make it easier.
So if there is no any question, then I would have a question to Teadora. My question is related to the planning of an experiment like single cell sequencing. What do you know? What is your experience? How long does it take to analyze the data from a single cell experiment? For example, if I have some regular sample from a brain, and I send it out to a service provider, they send me back the data. How long would it take me to create a publishable paper from it?
I actually never sent it to service provider for analysis, but what I can say, for example, the very basic understanding of the data would take maybe 1 or 2 weeks, but that doesn't necessarily include everything that you want to do with your data. So the more advanced analysis and everything that's maybe just initial looking at it, and maybe analyzing it properly could take months, or even more, depending on how experienced you are and how straightforward the data is. Because it also matters whether you know your sample very well, or whoever is doing the analysis knows the sample well. Because even in annotating the clusters properly, like identifying the subclusters, understanding the structure of the clusters, it's not always straightforward, not always trivial. And if you don't know the samples, don't know the target tissue, then it's even harder, because then you have to go and check out the literature. It could take longer than you expect.
Thank you. And I see a question from the chat. I think we'll try to unmute one participant.
Yes. This is Attila Mocsai from Department of Physiology. Can you hear me?
Yes.
Okay, great. Thanks a lot, Teadora. Sorry, I'm driving, so I cannot write messages in the chat. Thanks a lot for your nice presentation. I was wondering how much you have done, or are you aware of anyone having done such experiments in Budapest, in Hungary? How much experience is there if let's say if we want to do some single cell RNA sequencing, whom we should turn to, and these kinds of things. Can you give some information on that to us?
Well, I can certainly refer Tamás, the host of this talk. He has been working with single cell RNA sequencing for quite a lot of time, and I think he has good experience. As far as I see, in the last 2 years, this technique has started to become known in Hungary, but still it's very, very new. Regarding our work group, we worked with a Danish group who has good experience, and that's where we learned our experiences, and that's why we started to apply this technique. And as far as I know, there are some work groups in the Élettani Intézet of Semmelweis, and also starting out here in the Department of Anatomy. I think Gerendas Lily is also present. She is from a work group where they are just starting to apply single cell RNA sequencing. But there's not, I'm not sure that there's like 10 years of experience anywhere in Hungary. If there is, then it's with the collaboration of other work groups.
Because that would be very important. I think that the Department of Physiology group is actually our group. So we started to do some very basic trying. 10x we have discussed this with 10x, and we have started some single cell RNA sequencing just to have some idea. But eventually it went very slowly. We didn't know what to do either, and we didn't, even though they were very helpful, there was just not enough experience. So that's the reason why I also came to your talk, to the Zoom meeting.
In our case,
so
for example,
basically you're saying that there's not.
Sorry.
Yeah, go ahead.
No, I just wanted to say that in our case, for example, I am from a smaller work group with not so much funding, and we chose to actually just buy the whole service from a service provider. So from nuclei isolation, everything, and to do the single cell analysis by ourselves. So that's what we could afford, or try to cut the expenses. And so there are European companies with really good experience who can perform good single cell RNA sequencing and provide you with good data. And maybe that's also a possible solution, because I know that it can be very hard to and trying and expensive to try to optimize a protocol for your sample. But I don't know. Where did you experience it to be long? Or what posed as a challenge for you?
You know, we were just interested in this technique in general, and we wanted to work on do experiments on neutrophils, and the neutrophils are a unique cell type in that they are not very transcriptionally active, and they have their own challenges and limitations. But we certainly wanted to try live cells. So these were freshly isolated live cells. But just the how to sequence, how much to sequence, how to handle your data, your cells, your samples. Besides doing a zillion other experiments, there was just not enough motivation to go all the way through. That was the problem, and we felt that there was not someone whom we could turn to with all the small technical details that appear. So thank you very much. Maybe we come to you and approach you and ask
and try to figure it out.
Your experience. Okay, great. Thanks a lot. Thanks a lot, Teadora.
Thank you. Okay, I see we have another question. So the question regards having two different experiments. What can you use to compare the expression rates of given genes? So yeah, this is a more complicated topic. When you have different experiments, it's not like you can just merge them and analyze them together. As far as I know, so I'm not an expert either, but as far as I know, and what I used to do in this case is you need to have a special integration step in your analysis. Because if you just merge, for example, even if it's the same brain area, same types of cells, if you just merge different experiments, then you will have very major technical variance present, and the cells will just cluster based on their source. And you cannot really do anything with that. But there are, or there has been a lot of bioinformatical effort put into that problem. And so there are different approaches to reduce this batch effect. They call this batch effect. So I didn't want to go into so much detail of data analysis, but for example, the Syrah package, which is R-based, somewhere here, or Seurat, also has this step implemented. Integration is available, an available step of it. So you can tackle this problem, and you can analyze different experiments together in a, I would like to say, reliable way, but this is quite a fresh area of research. So you always have to be careful what you believe and what you don't believe. But together with your biological knowledge, it can be quite reliable. So to answer your question, it's not just certain parameters you use to compare the expression of given genes, but you have to apply a certain biological workflow or pipeline to make them comparable, but it's possible. I hope I answered your question, sort of.
All right, thank you very much. I think there are no more questions. If anything comes up in the future, you can reach out to Teadora anytime if you have her email address. And thank you very much. Thank you for everyone for the participation, and thank you, Teadora, for presenting today. It was a really good talk. Thank you very much. Bye.
Thank you for being here.